All right. Good morning, everybody. Happy Monday. So, as I was looking at this, I realized this chapter should have been last week. What happened? I must have just been having way too much fun. So, microbial genetics is what we want to think about. We will get on track here. Uh, there are lots of details when we think about genetics that ultimately we need to back it up and wait and say, wait a minute, microbial genetics, bacteria have shed tremendous light on mechanisms. And really, when we think about it, it makes sense. Bacteria are super easy to cultivate and to manipulate. And that in turn has opened up all sorts of potentials with our understanding of how these processes function across the board. So truly important when we think about well, everything that we're going to start spouting off here in just a second. So there is huge diversity when we think about a microbe compared to an elephant as our example. They appear tremendously different. Single-celled versus multicellular, prokaryotic versus eukaryotic, a lot of differences. But when we dive deep and start thinking about mechanisms, the genetics, a huge amount of similarity. DNA molecules the same, the assortment of those nucleotides dictating the respective proteins we produce. We may see some variation with that, but ultimately, the code that we think about is universal. All right. And with that, that opens up opportunities when we think about genetic engineering, when we think about biotechnology, the potentials to utilize things like microbes to our advantage are huge. All right. There are definitely some potentials, and we'll hit on this as we progress along. Prokaryotic and eukaryotic DNA, we do see some differences, circular for prokaryotes and linear chromosomes for eukaryotes. There's extra stuff going on in the DNA of eukaryotes. We'll talk about introns and exons. And we don't see that stuff in prokaryotes. It's all business, basically. All right. And so that creates one extra step when we think about trying to put eukaryotic DNA into a prokaryote. We got to manipulate it a little bit, and we'll hit that shortly. All right. So there's a lot of stuff that we truly want to think about as far as how these mechanisms are really going on. So initially, we want to think about the instructions in the first place. And we call this central dogma of molecular biology. This is the way stuff happens. All right. It's the flow of information. All right. So starting off with the instructions in the form of DNA, deoxyribonucleic acids, these are the instructions to basically build everything, right? Whatever it is that cell's doing. And a lot of those instructions truly are coding for proteins. We'll think a little bit about that stuff as we progress along. So in order to deal with these instructions, we've got to go through a couple of steps. All right. DNA and RNA molecules are different. All right. Double-stranded in the DNA versus single-stranded in the RNA. The sugars are different, deoxyribose and ribose. And thymines in DNA get replaced with uracils in RNA. So there's a bit of difference, all right? And it makes me think like maybe learning a different language, all right? Hello, Duolingo, right? Learning a different language, you might need some sort of translator. We may need to shift it from this to that. And that's ultimately really kind of what I think about with this. We need to go through this DNA message, this information that says, build me this protein, and we transcribe it into something that's readable by the rest of the cellular machinery. So transcription takes DNA and makes a copy of RNA, messenger RNA, ribosomal RNA, transfer RNA, boatloads of different types of RNA whose business is truly to build proteins. All right. So once that RNA, those RNAs get produced, then we want to start thinking about actually the building, taking those instructions and saying, I can build you this shelf or whatever it might be. All right. So translation reads that messenger RNA with the help of ribosomes and transfer RNAs, bringing in the respective amino acids. You get your protein. All right. As I deal with this stuff in biology, I like to think of it kind of like a cookbook analogy as well. DNA would be the cookbook, and all of the pages within that cookbook we could think of as chromosomes and instructions, genes, recipes in that cookbook that we ultimately might be able to utilize to make a cake or a cookie or whatever it is we want to make. As we read that, then I envision something like maybe you have some dinner and that recipe that you made was really good and the guests would like that recipe and you write it down on a recipe card. That's transcription, copying it from that cookbook onto the recipe card. And then they take it home and they read those instructions and build their own dinner. All right. Or make those cookies that grandma made that were so stinking tasty. All right. I mentioned that in a biology class, and I had an older couple that were in there, and she spouts off that grandma's cookies are so good because she puts a little spit in there. And I was like, oh, mental note. If she bakes me any cookies, decline, right? I know what you put in there. They might be the best cookies ever, but I don't think she really spits in them. But it caught me off guard. I was like, oh, is that what grandma really does? Right? So I would hope not. Now you're like, wait a minute, what does my grandma do? Right? So before we get going with all of the details of how we go about making our proteins, we need to think a little bit about our organism, whoever it is, our microbe, as we're in microbiology. The genotype, that's all of the instructions within that chromosome that bacteria harbors, right? That's all of the instructions. That's its genotype. All of the nucleotides, all right? But those genes that are on that chromosome, not all of them are expressed all of the time, right? It truly depends on the given circumstances. We could back this up the littlest bit and think about our own bodies and all of these chromosomes coding for everything that we need. But in nuclei within skin cells, specific genes are turned on. And in a kidney, other genes are turned on. And I'm pretty stinking sure you don't want kidneys growing on your skin, right? We turn some of that stuff off or flip it around and turn other stuff on, right? So it just depends. And there's lots of different ways that genes can be regulated. All right. So in this condition, we've got environment that's dictating specific things that may be turned on or turned off. All right. So dependent upon those specifics, we may see differing types of phenotypes. Our good friend E. coli loves the sugar lactose, and given the opportunity, it really likes that and ferments. You saw those tubes turn yellow, right? But if it's not in a habitat where lactose is present, do you think it would be wasting its time making all of the enzymes needed to break it down? That's pretty costly. It's going to shut that stuff off. We don't need it. There's no lactose present, right? So you'd have a different phenotype, different expression, right? We aren't using any lactose. We're not building any of it. Those genes are shut off, all right, or turned on, all right, dependent upon the specifics. And if the environment shift, we still have the instructions to be able to deal with those circumstances. That's important, right? So you're not just constantly pumping out lactase to break down lactose that's never even there, right? So there were lots of ideas about all sorts of stuff. If we go back to the 1900s, thinking DNA stuff, what is it? What is the instructions within these cells? Is it DNA? Is it protein? Those were things that we battled through in the first place. Watson and Crick showing us the structure of those nucleic acids, ultimately getting us to this point where like DNA is the instruction, right? So then there were thoughts of, well, how is it that the DNA replicates? And the middle, the semi-conservative process, this is the way that it happens. But there were other hypotheses as far as how this DNA molecule could replicate, all right? And they set up some experiments. They used nucleotides that had been radioactively tagged and allowed to witness what happens in the process of replication to shed light on what's truly going on here, all right? So as we look at this figure up at the top, all blue, these are all the parental strands of DNA, right? So anti-parallel, right? DNA strand, we're looking at two sides of our DNA molecule. We'll show some details of replication in a second, but the DNA unzips. We got a lot of players that get involved and they link up and start making complementary copies as those base pairs lay down. So the conservative idea, hypothesis, was that the parental strand serves as the template for the new DNA, the daughter strand, but it's not used. It just reads it and makes a copy, like you photocopied, all right? So you're left with your original untouched, if you will, and then you've got your new stuff, both sides of it being new, all right? And then through subsequent rounds of replication, we can see our parental still right here, and then we've got three others that are all new stuff, all right? I'm going to skip over the middle guy to the dispersive, all right? The conservative to me makes sense. That could be a potential. I told you semi-conservative is the real one. Dispersive, when I think about this, this seems to me to be the most unlikely hypothesis, all right? Could it be a potential? Sure, it could have, right? But it, to me, doesn't make a whole lot of sense. So the idea with dispersive is you unzip and you make new copies, but they're on both strands, and you get little bits of old and little bits of new. It's all smattered throughout there. Different, all right? You see the striped appearance as we look at the second generation. And then we get down to the third generation, and they all have stripes, if you will. Little bit of parental and a lot of new stuff. Varies, all right? The one that we know, all right? The semi-conservative process, both sides of that DNA unzip, and they serve as the templates. Molecules move down there, DNA polymerase that allows these nucleotides to come in and bond. It unzips, and we get one side red, one side blue, parental and new, all right? And as we go through this next round, we see our parental, and we see two copies here in the middle, it's all brand new, all right? Because this template, as we unzip, all red, now we get red side and another red side coming down. Same with this other inner middle, right? Giving us these two. And this one, blue and red, all right? And the same over here. And you see that repeating. And the next round, the second replication, the third generation to the fourth generation, you'd still have two light blues flanking all of this, but now you would have four double-stranded reds going on in between, right? So this is the process, and through radio labeling these nucleotides lent to our understanding that this is truly the mechanism, right? So this is basically that same idea, all right, but showing the actual experiment. So they used heavy nitrogen. We start off with our broth, our solution that these bacteria are going to grow in, and it's all heavy nitrogen. So as they're pulling in these nitrogens to build their nucleotides, we wind up with the sequences that are all radio labeled. They're all heavy nitrogen, all right? And once you've got all of that, then we take it into a new broth that's got regular nitrogen, light nitrogen. And as it gets incorporated, you go through and basically see what's happening. We can see with our conservative, doesn't. The dispersive does not. But the semi-conservative, as we move through here, shows this same idea. They use some centrifugation to spin these tubes down, and the heavy and light nitrogens will settle differently as it gets centrifuged. And then they can detect, as we see these bands, the position of that, and ultimately shedding light on this process being semi-conservative. We'll go through and see replication in a second. So this is an individual nucleotide. This is a guanosine triphosphate deoxyribonucleotide, all right? We call them DNTPs, all right? And these four different potentials. This is our variation: guanines, cytosines, thymines, and adenines, incorporated with the phosphates and your sugar, deoxyribose, all right? This is the base, all right? So the rung of your DNA ladder, the edge, is sugars and phosphates, and not the rungs are the nitrogenous bases. The backbone of it, excuse me, are the sugars and phosphates. And the rungs of the ladder are the nitrogenous bases. They will hydrogen bond with one another, all right? The backbone covalently bonds, sugars to phosphates covalently bonding, all right? And I'm pretty sure I mentioned before, these molecules, when they're coming into the mix, have three phosphates. Just like our ATP molecules that we think of as energy currency in the cell, those phosphates harbor a huge amount of energy. And it takes a lot of energy to get those backbones fused together. So two phosphates pop off, double dose of energy, and it fuses one phosphate to the adjacent sugar, all right? Building a very strong covalent bond, all right? That's the idea, all right? So as we move through here and think about this replication, it is rather complex, all right? When we look at up above, our one little piece of DNA, all right? In our bacteria, we've got a starting point, called the ORI site, or the origin of replication. All right. And this is where we're going to initiate DNA replication. All right. The DNA double-stranded unzips. All right. There's a lot of players involved with this, because if all we did was unzip and not keep it held apart, the likelihood of it coming back in hydrogen bonding right after we started to unzip is huge. All right. So lots of players. All right. As we move through here, we got a lot of different proteins, all different shaped as we're moving through here, that help to facilitate this process to get it to function like we want. All right. So initially, before we look at all of the stuff going on inside, we've got an enzyme called topoisomerase gyrase. This enzyme links up to the DNA that's twisted up, and it helps to untwist. So to make it a little more flat instead of coiled. All right. Topoisomerase does some untwisting. Our little triangle right here, this is like the business end of your zipper. All right. Whatever that's called. All right. That part of your zipper that does the zipping, all right, starts breaking those hydrogen bonds. All right. And so that's what helicase does, is it splits open that double helix, making it from double-stranded to single-stranded. Now we're open for business, if you will. Both strands are accessible now. They're not hydrogen bonded. Our little tannish guys right in here, these are called single-strand binding proteins. Once we make that break and unzip, these proteins link up on opposite sides to keep it from coming back in hydrogen bond and back upon itself. Now we're open and ready to do stuff. All right. So all of these get started, untwist, unzip, and hold it open. All right. With those binding proteins. All right. After that, you need to start laying down the groundwork for the DNA polymerase to get in there. When I think about this, it makes me think of like an interstate. And if you want to get on to the interstate and drive yourself up to Idaho Falls or wherever, right, you use an on-ramp, right? That on-ramp gets you onto the interstate, and then you're good to go. All right. So RNA primers lay down a chunk of RNA molecules that complementary pair to the DNA, right? But that RNA, it's the on-ramp, right? It's what allows DNA polymerase to recognize and get on to the interstate, the DNA interstate, all right? So RNA primers, and we're not seeing our green guy up here, but it was, all right? It started up here, all right, and laid down that little bit of on-ramp, and then we start going, all right? As we look at this, we've got two different strands, different orientations, forward and reverse, all right? Leading strand is the proper orientation. You're going in drive, all right? The lagging strand is the opposite orientation, and our primers, our polymerases, excuse me, they don't like that. If you wanted to go to Idaho Falls and go in drive, you could go 80 miles an hour if your car will let you, right? You could go 80 miles an hour, right? But imagine getting on the interstate and deciding, I'm going to just go in reverse, right? You think you could go 80 miles an hour down the interstate in reverse? I bet not, right? I bet you your car will not let that happen. You might be able to get up to 25, all right? But I bet you you're not going to go 30 or 80, right? 30, yep, can't do it, all right? I was saying this exact same thing, all right, in a biology class, talking a little bit about forward and reverse type of stuff. And then after school, I found myself going to Costco. The back road next to the railroad tracks, you may know where that is. It was the secret way to get there till everybody's like, wait a minute, this is the best way to go, right? If you don't know that, now you do, right? So I'm driving on this back road to get to Costco, and a car coming up on me is going in reverse. They're driving in reverse. They're in the right lane, right? But they're going in reverse. And then there's another car driving. They're going the right way. It's some high school kids, all right, is what I figured. And the ones that were going the right direction were filming the other people going the wrong direction, but on the right lane, right? And I could see as I'm driving, and they all seem like they're having a good time. And I'm like, I'm pretty sure someone's going to crash here in a second. Glad I got through there, right? So I can get my cheese. Interesting, but only about 25, right? And I was impressed with their ability to be driving backward like that. Not going to try it, but impressed. All right. All of a sudden I'll get these videos. Look what I did after class. I drove backward down to the sub, right? Took out a couple pedestrians. It's okay. All right. So going in reverse takes a little bit more. All right. A little more focus, right? So these going backward, it's little bits, little stepwise approaches, all right? And we do it in these increments that we call Okazaki fragments, all right? It was Japanese scientists that ultimately recognized this process of forward and reverse and it being different, all right? So in order to go backward, it results in numerous RNA primers that get laid down, giving the opportunity for the DNA polymerase to get in there and make a little bit, all right? That causes an issue, all right? We should have been in English in the first place. All right. So the green guys, the RNA, all right, we don't want RNA left in our DNA. That's a problem. So DNA polymerase 3 is the one that's actually making these strands. DNA polymerase 1, a different polymerase, it's more of a proofreader. It's checking for issues, and it's also looking for RNA. And when it comes across as it's done its process, DNA polymerase 1 goes after, shortly after, and is checking stuff. And any place where it sees the green stuff, the RNA, slows it down, chews it off, and puts a DNA nucleotide in its place. So it's like the road repair crew fixing issues in there. So it's nice and smooth, right? Polymerase 1 goes through and fills all of that in. And then there's issues with this because once we fix that last little bit, we see a gap here, right? This sugar phosphate and this sugar over here, we're not covalently bonded together. We already laid this new piece down that was DNA, and we just chopped out the green guy, right? So that little break in the DNA, another enzyme called DNA ligase comes through, and it's really the one that fills in the pothole, all right? It's the one that causes a covalent bond to form between the two adjacent nucleotides, one of which used to be RNA, all right? So DNA ligase is like molecular superglue, all right? It causes a covalent bond to form where it didn't have the opportunity to do that, all right? So a lot of different things happening. DNA polymerase 1 goes across the top bit as well, proofreading, ensuring everything happened. Polymerase 3, that's bringing in the nucleotides, it does a bit of proofreading as well, looking for mismatch sort of stuff, and it can correct it pretty proficiently, all right? But not 100%. And polymerase 1 is the backup, all right? Like writing a paper and then maybe having somebody else read through it just to check and see, does this make sense? Or writing it, not the same night it's due, writing it a couple of days before, putting it away, and then pulling it back out and reading it before you actually submit it. Because sometimes you're like, wait a minute, this sentence makes absolutely no sense. Or here's a typo or something like that that you wouldn't have caught as soon as you powered through there at 11:59 and it's due at midnight, right? You may not catch that crap, but if you give yourself a little time or some other pair of eyes looking through it, a proofreader, then they may catch it, right? It's important, all right? So one thing that I will point out, when we're looking at this, we're showing you, just as you see this little box up here, we're showing you one half of it, all right? And at this origin site, on our top bit, we're in the right orientation, and we call that the leading strand. But if you look to the other side of it, at this bit of the origin of replication, this is backward, but we're on the same strand that was forward, right? As we go to the opposite side of that origin, and we're going to replicate in both directions. It's bidirectional replication. All of this same stuff is over here too, all right? So as we are unzipping and moving in both directions around this rubber band of a chromosome, one part of it is in the proper orientation, and on the exact same strand opposite of that origin, this is now the lagging strand, and it's doing exactly what we're seeing right here. And down below, this happens to be leading as we are moving toward the zipper, all right? And this away from it, all right? So different, all right? And this lends to a bit more confusion when you try to look at this as the whole thing. It's a lot easier to look at it half, all right? And then just remember, on the opposite side, we're going to flip this upside down and see the lagging on the top and the leading down below. Same idea, all right? Just depends on that orientation. So here, as we look at this, eukaryotic chromosomes, linear, all right? And there's multiple origins of replication. Instead of a single ORI site in our prokaryotic chromosome, eukaryotic chromosomes have a handful of ORI sites, and it's one long strand. So we can start replicating multiple points on that chromosome and start this process simultaneously to speed up this process, all right? And this, if we're thinking, you know, in a eukaryote, this is happening in the synthesis phase of our cell cycle, right? While we're still hanging out in interphase, right? You replicate really very rapidly, all right? So we can see it's the same idea as this, just multiple bubbles, if you will, and then we will meet, done deal, right? And when it's all said and done, you've got two new strands and the parental strand, semi-conservative process, all right, as we move through there. And like I said, bacteria, single origin, multiple, single, all right? So this is also something that we see in eukaryotes. You don't see it in prokaryotes, all right? So with those linear chromosomes, as we go through and replicate, this has to go back to thinking about the lagging strand, all right? Where RNA primers lay down a little bit and we go. As we start doing this, toward the end of your chromosome, technically there's not a spot for that to happen, and you get an overhang. That's a problem, all right? DNA telomerase can make a copy of that. And now that overhang is no longer an overhang, it's complementary base paired. all right? Typically, our cells aren't harboring telomerase, all right? There are some that do this. I'm thinking stuff like stem cells. There's telomerase activity, all right? But most of the time, not. And that ultimately is the aging process. As we go through rounds of replication and start breaking off pieces, these tails that don't get base paired, we start degrading those chromosomes. At the end of the chromosome is a huge amount, a big long sequence of repeating nucleotides that we call telomeres. And the telomeres are like caps, protective mechanisms on those chromosomes to prevent that breakage from actually hitting instructions, all right? We can lose this stuff because it's not coding for anything. It's there to get lost, all right? And as we go through numerous rounds of replication, you get to the point that we've run out of telomeric DNA, and those cells are going to start to die, all right? And ultimately, so will you, all right? Having telomerase could be, have we just found the fountain of youth? Cancerous cells have telomerase activity. I wouldn't call that a fountain of youth, all right? That's a different situation. And every time they replicate, telomerase goes through and now they behave as if they are a brand new chunk of DNA. And they keep replicating over and over, always fresh, if you will, all right? So that's the idea of what's happening with telomerase. And like I said, it in normal situations isn't functioning, all right? But then you're like, maybe we would like that, right? So with our circular chromosome, back to prokaryotes, all right? As we see this process of replicating, we call it a rolling circle, all right? As we make new copies, all right? So a nick, we break it, our polymerase 3, that's going to be bringing in these nucleotides, starts moving through, and again, lagging and leading, all of that stuff's happening. We're just not showing that detail, all right? So you can see the new in red getting laid down, and as it's all said and done, 2 copies, all right? 2 cells, if you will, all right? So shifting, we see our strand of DNA, all right? And now we've got RNA polymerase here in the middle. This is a different polymerase than the one that's going to be laying down the on-ramp, all right, for synthesizing DNA. This RNA polymerase is making a copy, all right? So this is transcription that we're looking at, all right? Our RNA polymerase reads one strand of that DNA and complementary base pairs the RNA nucleotides together. They hydrogen bond briefly, and we fuse their ribose phosphate backbone. We fuse it together. So we get a sequence of messenger RNA, all right, that comes out of here. As we look at it, the 2 copies of DNA, up at the top, we call this the non-template DNA strand, and down below is the template, all right? The template strand going 3' to 5', and our RNA getting produced in the 5 to 3 direction. This is the way we want it to go. This is forward, this is drive, all right? So RNA is made 5' to 3'. As we read the DNA, 3' to 5'. The DNA up above is in the opposite orientation. But if you look at, and we've got some stuff missing in here, but if you look at it, the RNA that we've made, except for uracils switched with thymines, it's a mirror image of this non-template strand, all right? This RNA looks exactly the same as this, all right? And this piece says that's what I want to have made, all right? So we call this the template strand, but really this is the one this is what's saying it, right? And we translate it into the code that the rest of the stuff needs. The uracils are how we read it, right? So 3' to 5' DNA, 5 to 3 with the RNA, and now we've made the messenger RNA. So all of these bits of nucleotides, we look at them in threes, all right? We also went through and figured out how all of this works, and we call these things codons, all right? The idea with this, and this is an amino acid table, I'm assuming you have probably seen this at some point in your life, likely biology, right? So the way this table works are 3 base pairs, the first base pair, the second base pair, and the third base pair, all right? So I always go with leucines. If we start off with our first one, it's a uracil. Our second one is a uracil. And then over here, it's either an A or a G, all right? And either of these gives us a leucine, all right? Adenine or a guanine, all right? Both of these two, adenines and guanines, are purines, and the cytosines and thymines are pyrimidines, and purines complementary pair hydrogen bond with pyrimidines, purine, pyrimidine bonding together, all right? So leucine up above, we got 2 different ways to build leucine. But then if you look below, there's another leucine, right? And this one gives us 4 potentials. If you say Cu, we don't care what you say in your third position. We're going to bring in a leucine. That amino acid gets deposited in this growing polypeptide chain. We call this redundancy. There's more than one way to say this word, all right? To say whatever it is, to say leucine, all right? And we've got 2 ultimately different ways that we may be saying leucine, all right? Transfer RNAs look at these codons, all right? And the transfer RNAs, we say they have some wobble. The tRNA can wobble on the back end, the third position, but you don't get any wobble on the front, all right? So one transfer RNA recognizes Cu, and it knows I'm bringing in a leucine because it doesn't care, all right? But up above, with this phenylalanine and this leucine combo, if you say UU, I'm not just going to bring in a leucine because maybe I needed a phenylalanine. These tRNAs want to know UU purine or UU pyrimidine, all right? We don't care if it's an A or a G. As long as it's a purine, we're good to go and we're bringing in a leucine, or oppositely, a U and a C for the phenylalanine, all right? So 2 different transfer RNAs for this, all right? Some of them, only a single AUG brings in a methionine. Colored it in green because we call these guys the start codons. Every protein sequence begins with AUG, begins with the methionine. Doesn't mean that every single protein has that as the final form, because there's a lot of modifications that might happen. We call them post-translational modifications. Sometimes those big long polypeptide sequences get cut in half, or we may chop off the front bit of it. It may not all start with the methionine, but when we initiate everything, it does. All right. There's another one, tryptophan up here. It's another one that only has a single codon to build this. All right. Everybody else has multiple ways, but tryptophan and methionine, single way. All right. In red, we've got our stop codons. All right. These don't code for an amino acid, maybe a release factor to get those ribosomes to separate off, but I think of it as we look at these 3-letter codons, it makes me think of little kids first learning how to read. And when you're doing that stuff, it's like 3-letter words that we're learning, and we're doing real simple stuff. All right. But if you're learning that, the first letter in that sentence is capitalized. Here's our capital. And as you finish that sentence, you punctuate it. And we could do a period, question mark, or an exclamation point, right? 3 ways to punctuate that. And fortunately for us, 3 ways to stop this. It seems to fit with that little kid book, all right? Spelling out something, all right? And a couple of different ways that we may spell that word through some simple mutations. You may have some base pair that's different, but if it codes for the same thing, we're in business. Doesn't matter, all right? So this also is a mechanism that helps to ensure issues like that maybe don't become big problems, all right? Where you've got more than one way to code for the same thing. There are lots of times with bacteria, there are looking at sequences and looking at proteins that are being produced. There might be specific codons that are preferred with that organism, and we call that codon usage bias. So if they tend to always, any of these could make leucine, but my good friend E. coli always uses CUG, right? Maybe, maybe not. I'm just making that up, all right? But something like that, we would call that a codon usage bias, and you would see it's all like this or almost all like that, all right? And others may do something else, CUC, right? And that may allow us a little bit of detail to get, an understanding about variation within prokaryotes, all right? When we see changes in these nucleotides that aren't the coding for the same thing, well, now you've got a mutation that's not silent, all right? A silent one codes for the same thing. Now you've got a mutation that codes for somebody completely different. Now, instead of slapping an alanine in there, we get a threonine, all right? Or a proline, right? Not what we wanted. And all of these amino acids have different characteristics: acidic or basic, positive, neutral, negative, whatever, right? They may fold or twist as they participate with other adjacent amino acids. And now you slap somebody else in there, and the way it folds or twists likely is going to be completely different, right? Or maybe you get a mutation and you wind up with a stop codon where you didn't want it. That's a problem, all right? That's a bigger problem, probably, all right? So with our prokaryotes, not having all of the extra stuff going on in the chromosome of a eukaryote, introns that have to get dealt with, we can start making messenger RNA, and we can start building protein almost simultaneously, all right? So in this case, we're one polymerase that we're seeing right here starting to make messenger RNA. Before we've ever even finished making that message, we were so stinking excited to make these cookies. I started using the recipe before grandma ever finished writing it, and I'm already making the cookies, all right? That's what's happening right here. We're building it before we're done saying. This is how everything I need. And technically, multiple ribosomes can be linking up onto this message. Now we've got an assembly line going on, because not only did I like those cookies, I brought them for everybody in here. And now every single one of us simultaneously is making these cookies, right? That's a lot of stinking cookies. Bake sale, right? I can feel it. So we can produce boatloads of message, because we could have multiple messenger RNAs starting to be created, and we also can have multiple ribosomes linking up, and we start pumping out huge amounts, start pumping out huge amounts of product, protein, whatever it is we want it to be building. All right. That's the idea with this. And it can be very quick, very proficient. All right. And in eukaryotes, not like this. We've got other stuff going on. And that messenger RNA gets produced in the nucleus, and then it's got to get exported out of the nucleus. But before we do that, we've got to process it. We got to chop out all of the introns, all of the stuff that we might think of or maybe called at one point junk DNA, stuff that's not needed. That's got to get removed. And we've got to push all of the business instruction together, all right? And then we leave the nucleus and associate with ribosomes, either free floating in the cytoplasm or studded on the rough endoplasmic reticulum, just depends on what we've got, all right? And with that, there's a little bit of other sequence that gets laid down onto the eukaryotic message that's like a zip code. If you want to write a letter, an actual real letter, and send it to somebody, you put their address on there, and you have to have a zip code so they know where's it going. The messenger RNA has a little bit of zip code, if you will, and as it's leaving the nucleus, that code basically says, I'm going to the rough ER or I'm headed to the cytoplasm. It knows which ribosomes it should be associating with and where those proteins are going to go, all right? That's pretty impressive, I think. So our tRNAs, when we think of these, this is like, they describe it like the second code, all right? The DNA has the instruction. We build that copy in messenger RNA, and then the tRNA, it's going to complementary pair up with that message. And if it's anticodon, the opposite of the codon, complementary pairing, if it's anticodon makes contact with the codon, we're the right one. But if that tRNA comes into the ribosome and it doesn't have the right code, it's kicked out of there. We move through there till the right one comes in and we keep building, all right? So the tRNA or transfer RNA is single-stranded, although there are parts that look double-stranded. It's not, all right? It folds back on itself, all right? There's a part at the top. As soon as I hit this, I'll be done. I swear it. A part at the top where the amino acid attaches, all right? And a part at the bottom where they link up to the message, all right? The attachment site and the binding site. And if all of this works, we're going to transfer those amino acids to a growing protein chain, all right? That's the idea. Where's Dr. Meldrum at? I was sure he'd be popping in here and be like, you're going too long. All right, I will quit with that. We'll pick up with the rest of this Wednesday. Farewell, good day.